So I have the pleasure of introducing Dr. Miao, who is our third grant recipient. Dr. Miao is an instructor in the Bluebird Circle Development Neurogenetics Laboratory of Neurology at Baylor College of Medicine. He's been studying Cacnata related neurological diseases since 2015. His research is devoted to understanding the molecular and circuit mechanisms of neurological disorders, developing novel treatments for neurological disorders and improving neural functions, developing new diagnostic and research tools for early diagnosis of neurological diseases. Dr. Miao obtained his PhD from Shanghai Institutes for Biological Sciences, University of Chinese Academy of Sciences in 2014. And with that, I turn it over to you, Dr. Miao, with the utmost gratitude for the work that you are doing.
Thank you. So good morning, everyone. Thank you for creating the Cac1A Foundation, and thanks for organizing this conference to bring us together. Also, I sincerely appreciate those who generously donated to the foundation, which provides a strong support to my research. It is an honor for me to have this opportunity to share my thoughts and research with you today. Here is a slide from research collection at Karolinska Institute with sections from human cerebellum. Research has written, Gift of C. Golgi to G. Research, 1886. If you look at it at higher magnification, you will see the dendritic ramification of Purkinje cells still clearly visible after more than 100 years. They look like bushes. The Purkinje cells are some of the largest neurons in human brain. The revelation of this subcellular structure was made possible by a staining technique named Golgi staining. The technique was discovered in the late 19th century by Camillo Golgi, who was an Italian biologist and pathologist. With this technique, scientists examined the structural development of Purkinje cell by staining brain at different developmental stages. Here are Purkinje cells from newborn, young, and adult cat brains, respectively. This developmental process is comparable to the growth of a tree in terms of more and more complicated branches until maturation. Similar course is taken by human brain. Neurodevelopmental diseases could appear at early stages, even just after birth, which might make it hard to cure them at later stages. Early diagnosis and intervention would be necessary. To learn how brain works and why some doesn't work properly, EEG technique was developed to monitor its electrical activities in early 20th century, particularly in epilepsy patients. These twin girls, Constance and Catherine, they started to have seizures at 6 years of age. 5 years later, which is 1949, they saw Dr. William Lennox, who was a pioneer in EEG technique. EEG recordings on them showed that they both had absence seizures, manifested as spike wave discharges on their EEG, showing abnormal electrical activities in their brain during seizures. However, the genetic variant or variants responsible for their disease was unknown. To study the genetic, cellular, and circuit mechanisms underlying various diseases, mouse is used as a model organism, not only for its similar brain development like human, but also because scientists have the ability to manipulate mouse DNA to produce transgenic animals, where specific genes can be modified or even removed in order to study their functions in health and disease. In 1957, an abnormal wobbly gait was observed in a litter of mice in Jackson Memorial Laboratory. Due to their wobbly, see these mice, it walked pretty wobbly. Due to their wobbly, obvious motor dysfunction, they were identified and studied carefully. A recessive mutation in their genome was identified to be responsible for their behavioral phenotype and was named Tottering. As Dr. Nobles introduced yesterday, more than 40 years ago, he implanted EEG wires on Tottering mice, and surprisingly, he found that they showed abnormal bursts of bilateral synchronous spike waves accompanied by behavioral absence attacks. It replicates to a large degree the absence seizures in human patients. It was the Tottering mice was the first genetic mouse model for absence epilepsy. Tottering mice is the best characterized model of the CAC1A mouse mutants. This CAC1A point mutated variants renders the PQ channel with loss of function, that is a reduced Hessian current density compared with wild-type animals, with no significant change in their single channel conductance. Interestingly, inborn errors in CAC1A do not lead to absence seizures in Tottering mice at birth. They instead show a delayed epileptogenesis during postnatal development, which correlates with the expression of CAC1A in the brain. As you see, CAC1A is more and more expressed in the brain, particularly in the cerebellum and hippocampus. And also it correlates with single cell structure of Purkinje cells. As an example, it also coincides with the development of various brain functions. The development of sensory and cognitive skills is highly plastic, both in animals and in human beings at early stages. These windows are named as critical periods. Normal development in these periods is essential for the emergence of proper brain functions. It is unknown whether neurogenetic disorders emerge from developmental deficits in these periods. It would last require early therapeutic intervention to cure the disease. Or is it, the major question we want to address is it ever too late to cure the disease? Animals with inborn genetic defects will have the disease. Specifically, we want to understand, do absence epilepsy and ataxia emerge from developmental deficits that would require early therapeutic intervention, or can they also arise in a mature network? If early deletion is necessary for neurological phenotypes, then treating channel deficits at the later stages might not be effective. To answer this, we created a later onset deletion model. With this tool, gene of interest could be deleted while a chemical trigger applied at any time. So this is an inducible knockout strategy. Specifically, CAC1A engineered mice, here named as LOXP mice, were crossed with an inducible Cree mouse line. And the offsprings, some of its offsprings in these mice, CAC1A could be deleted upon tamoxifen injection at any time. And the LOXP littermates were used as controls. So here are the mice that we generated So before induction, both animals had normal behaviors, as you couldn't distinguish which one is mutant and which one is control LOXP mice. Now I think anyone can tell which one is mutant and which one is control litmates after tamoxifen injection, because for this mutant, it displayed motor dysfunction deficits, just like Tottering mice. It is pretty ataxic, but not its control LOXP litmates. To quantitatively measure its motor function, standard rota rod test was performed. So for both mutant and control mice was placed on rota rod. So mutant mice fell from rota rod much earlier compared with control mice. So we measure the latency to fall from rota rod. So you can see mutant mice has a shorter latency to fall from rota rod. So to confirm that Cac1a is indeed deleted in the brain, an immunostained Cac1a in brain sections from control and mutant mice. So for a normal brain, Cac1a is almost expressed everywhere. In particular, it is highly expressed in the cerebellum Purkinje cell. You can see it is brighter in cerebellum. While in Cac1a deleted mice, Cac1a expression is largely reduced. These boxed areas are further imaged at higher resolution. So you can see Cac1a is ablated throughout the brain in Cac1a deleted mice compared with normal mice, including cerebellum Purkinje cell, thalamus, hippocampus, and the cerebral cortex. So we also monitored EEG activities before and several time points after Cac1a deletion. So mice before Cac1a deletion, they had indistinguishable EEG with control litmates. Well, within 2 weeks after injection, they started to have absence seizure, and the seizure become more and more frequent and persisted longer. Here is an example absence seizure observed in mutant mice. So please pay attention to the right arrows. Sorry, my screen is free. Started. Seizure ended. So you can see the absence seizure is accompanied by behavioral arrest, and the mice continued moving around after the seizure ended. So in summary, we found that adult loss of PQ channel results in ataxia, and adult loss of PQ channel also could induce absence epilepsy syndrome. These results suggest there is no critical period of Cac1a related diseases, and ataxia and absence seizure dependent on adult PQ channel functions. These results also implicate that it's probably never too late to cure the disease, and these diseases are not dependent on developmental deficits. So we are thinking about how we can cure these diseases. One strategy that we use is trying to enhance PQ channel function to treat diseases caused by loss of function variants. And as far as I know, Roscovitine is the only one PQ channel potentiator reported up to date, and it is a pretty promising one. It can not only reversibly enhance PQ channel currents in cultured neurons in a dosage-dependent manner, but also it has been evaluated in several phase 1 and phase 2 clinical studies and has shown early signs of anti-cancer activity in approximately 240 patients. In addition, these clinical trials have demonstrated that it is safe and well tolerated by both healthy volunteers and patients with various diseases. It has been evaluated as potential drug to treat cancers, neurodegenerative diseases, inflammation, viral infections, polycystic kidney diseases, and glomerulonephritis. So we want to determine whether roscovitine as a PQ channel regulator can alleviate epilepsy and ataxia in the tottering mice. However, before doing that, several points need to be considered for preclinical studies with roscovitine treatment in mice, including its dosage. So how much roscovitine could enter the brain? And second, potential different effects on wildtype and PQ channel variants, as usually patients carry PQ channel variants. Well, the roscovitine was found to be effective in wildtype animals, so it doesn't necessarily to be as potent as on wildtype channels. And thirdly is chemical purity, as some scientists reported that commercially available roscovitine might not be very pure, and those not pure roscovitine might have different effect on PQ channel. And finally, roscovitine, the chronic effects has not been tested before. So we take a 4-step process for preclinical study of roscovitine treatment, including dosage and safety evaluation, effectiveness assessment. And if it is not effective at a lower dosage, we would further escalate dosages to a higher dosage. And then we could probably draw some conclusion on our studies. As a first step, we have already evaluated the safety at a dosage of 3 mg per kg, which was used in a published paper to using roscovitine to treat stroke. And we found that it was well tolerated by mice. We did not find any significant differences between EEG before and after treatment. This is before treatment, this is 30 minutes after treatment, and this is an hour after treatment. And we will further tested in Tottering mutants. Finally, I hope that this study would pave a way to establish a safe and effective treatment for diseases with PQ loss of function mutations. And it would be impossible without these funding supports. Thank you.
Thank you, Dr. Miao. And we now do have time for questions if anyone has any. I can start with one question, and you are welcome to use the raise your hand function or enter your question in the chat box. But Dr. Miao, just to help us, those of us who I guess are less entrenched in the research process, understand, if you get promising results on a mouse, what is the next step generally to get that then for humans to use, to determine whether it's going to work in humans?
So if it is promising, usually it would go into like a complete clinical study, including 4 steps that I introduced previously. First, it needs to be evaluated whether it is safe in human patients, in a small group of human patients. then it would further be evaluated whether it is effective in human patients. Then it could be expanded to phase 2. I think the effectiveness will be evaluated in phase 2 clinical study in a small group of patients. And then if it is proved to be effective, then it would be expanded to a larger group of patients. Since we just, I think many of you might heard a lot of news about like COVID-19 vaccine information, how it was developed. So usually it would take at least 3 phases of clinical trials. So basically we are doing something similar, but here we just use mice as our patients.
Dr. Miao, what does the timeline usually look like for those sorts of things? And I know that it varies greatly.
As I'm a basic scientist, so I'm not that familiar with clinical studies, but usually it could take a very long time. I mean, it depends on how efficient like the clinical study can recruit the patients. So I think it is one of the benefits that you organize such kind of Cacnata foundation. So a lot of patients could know the resources as soon as possible. So could possibly be enrolled in those kind of studies. That would speed up the clinical studies. And also many things should be taken into consideration in terms of safety and effectiveness. I mean, how long those patients should be evaluated, not just 1 day, 1 month, probably several years to draw a solid conclusion whether it is indeed helpful or not, or whether it had any severe side effect so that it should be withdrawn or like go into further I mean test. Yeah, so it could take a long time, but as long as it is positive, it's still quite promising, and we can go forward step by step. And finally,
Thank you. And yes, you're right. That is one of our main goals, is to make sure that as there are clinicals available, we get that out to our community. Question from Roger Bannister. Will you assay the effects of more specific Roscoe derivatives such as GB58?
Add one note for the previous question. So it might be hard to like, it takes time and a lot of efforts to test just one drug in patients. So the advantage of study in laboratory is that we can simultaneously test the different strategy approaches in the lab, in particular based on the findings from our studies, meaning the mechanisms underlying the diseases. So we can parallelly test different kind of treating strategies and probably select the most promising one to go into the clinical studies. Yeah.
Thank you. And then we did just have a follow-up, just a comment from Roger Bannister that there are a number of higher affinity compounds which are derived from Roscoe, and these may also be of interest. So thank you for that comment.
Thank you. That's a very good point. As Roscovitine is not selective to PQ channel, it also targets some other proteins like including other kinases, CDK, etc. So if there is some like alternative chemical that is more specific to PQ channel, then it's likely that it might have less side effect compared with Roscovitine itself. So that's a good point.
Thank you.
Thank you.
Thank you, Dr. Miao.
Thank you.